the devastating rice blast fungus, Magnaporta oryzae, also as you've heard, also known as Prerequisites oryzae. And what I'm going to try and do is really answer these two questions. So how does the blast fungus infect intact host plants? So how does it get into a plant? And then once it's inside a plant, how does it move between cells and how is it invading plant tissue? So those are the things I'm going to set out to try and explore in today's talk. So rice blast is tremendously important, as many of you indeed will be aware from first-hand experience of trying to control this disease. So rice blast causes these ellipsoid lesions on the surface of seedlings. So in very heavy infections, it will kill seedlings completely, but also it will spread to other above ground parts of the plant, including the neck and panicle. And it's really neck blast and panicle blast that are the most devastating forms of blast in terms of yield reduction, and the one that therefore the farmers fear the most. And you can see this sporulation around a node, and of course what will happen then is that the inflorescence carrying rice grain, the panicle, will fall away and will rot away. So there'll be, and there's also coupled with less seed set. So this will cause really substantial yield losses. And in fact, this I took this picture several years ago in Hunan province in China, and the farmer there was facing around 80% yield losses. So it's a really important disease wherever rice is grown in the world. So every type of ecosystem, every type of rice is affected, but particularly rain-fed upland rice and rain-fed lowland and upland rice, I guess, are the ones which are the most seriously affected by blast. But it can happen really in under any of the cultivation methods, as you'll be aware. But the fungus itself is able to exist as multiple host-limited forms. In fact, the fungus, Magnaporta oryzae, the species, can infect more than 50 different species of grass. It can affect many forage grasses, for example, lolium, perennial ryegrass. It can affect many other crops, such as the eleusines, so finger millet, and indeed it can affect pearl millet too. So a whole variety of different grasses and millets are affected by rice blast. But generally, what this the way this works is that the individual fungal isolates that infect different species of grass are only able to infect those species of grass and none of the others. But occasionally, the fungus will undergo host jumps. It will move from infecting one host to another. And recently, the fungus made a host jump from species which were affecting grasses such as lolium onto wheat. And this happened in South America, first of all, in around 1985. And wheat blast disease occurred, Brusone disease, and it was restricted really to El Nino years under those climatic conditions in South America. But it spread quite rapidly across South America into indeed Bolivia and Paraguay and into Argentina. So it spread within South America. But something very serious happened in 2016 because wheat blast was first shown to have moved out of South America, and it moved into Bangladesh. And this is indeed in Mirpur, which is a province which is bordering West Bengal. So it's very close to the Indian border. And I visited in 2019 and saw firsthand the devastation caused by wheat blast. Wheat blast causes this bleaching disease, which can lead to complete yield loss. So you see whole fields which turn white because of the bleaching caused by the fungus as it infects these spikes. And it really was absolutely devastating. In the first year, it destroyed about 15,000 hectares of wheat. And in every year since, there's been wheat blast outbreaks in Bangladesh. And you can see that the way that the disease manifests itself, these bleached heads, and then a ring of sporulation, just resembling the neck blast symptoms that you see for rice blast. Now, if that wasn't bad enough, the disease then turned up in Zambia, in sub-Saharan Africa, and it was reported in 2020. So this devastating fungus now threatening wheat production in Zambia. There are reports in neighboring countries to Zambia that wheat blast may be present again. And recently, we've been working on trying to understand exactly what's been happening. And in fact, yesterday, we submitted a preprint which suggests that there's actually a pandemic clonal lineage of the wheat blast fungus, which is spreading around the world. And it's the same clonal lineage in Zambia as the one that's in Bangladesh. So it's actually probably a single clonal lineage has actually spread from South America to 2 other continents, to Asia and to sub-Saharan Africa now, and therefore it's going to be sort of incumbent on all of us to do all we can to limit the spread of this disease, because clearly it's a really serious concern given the wheat production in India, for example, that this disease is not allowed to spread any further. So this is a really serious situation. The other thing that's worth saying is that wheat blast and rice blast are now in the same location, and that's never happened before because the host jump came from a grass species. So there's now the opportunity for interspecific hybrids between wheat blast and rice blast, which is again a really serious concern. So that, by means of introduction, shows that rice blast is an incredibly serious disease of rice, but there are other diseases caused by blast, notably wheat blast, but also finger millet blast, which are also extremely serious and threaten global food security. The thing that really interests me the most about rice blast, though, really is the way in which the fungus is able to gain entry to plants. And this is the life cycle of rice blast. You can see the fungus sets out this germ tube, which germinates from the 3-celled spore. That leads to production of a special cell called an Appressorium, which penetrates rice plants, and then the fungus is able to rapidly colonize plant tissue. And I'm going to talk you through this life cycle for most of the talk today and tell you about the different aspects of the biology. But in general terms, what I want to really say about this is that what we can see is the fungus is able to switch between polarized growth and radially symmetric non-polarized growth. And in very fundamental cell biological terms, that's what's happening. There is continual transitions between polarized and non-polarized growth. And the ability to regulate that is fundamental to being able to cause blast disease. So how does the fungus infect host plants? Well, first of all, it germinates. And here's the spore germinating. This is a time-lapse movie. And you can see that dome-shaped cell on the right forming the Appressorium here. And you can see it becoming very dark at its edge. And that's melanin building up inside the cell wall. So the Appressoria become pressurized and they build up melanin in their cell walls, which enables them to maintain this pressure, which they can then apply at the surface of the rice leaf. I hope that what you can also see is the spore, the 3-celled spore, collapses during this whole process. So that by the end of this process, what you see is this on the rice leaf surface. Here's the Appressorium, which has developed pressure, and the spore is now an empty bag. It's just an empty bag. All of its contents have been recycled into the Appressorium. So, and that happens every time that the fungus infects a rice leaf. So how does the Appressorium work then? So here's the dome-shaped Appressorium again on the waxy rice cuticle. And what happens is that glycerol accumulates to very high concentrations inside the cell. That leads to osmotic potential, osmotic force, osmotic pressure, because water will rush in against that concentration gradient. So that's what's generating the hydrostatic turgor. That leads to really enormous pressure, 8 megapascals of pressure, so 40 times a car tire, so 80 atmosphere. So really high pressure is developed inside the cell. And that's then applied as protrusive force at the base of the Appressorium, enabling a very narrow penetration hypha to rupture the cuticle and gain entry to the leaf. And we know that the fungus will break thin plastic membranes. It's able to physically break surfaces. So although it probably uses enzymes to aid its attachment and to aid in the breakdown of the cell wall, it doesn't absolutely need those. It can do this simply by using physical force. So what are the prerequisites for making an Appressorium? Well, one of the prerequisites is that the fungus has to be able to perceive the surface characteristics, so the surface hardness and the surface hydrophobicity, and also chemical signals from the plant. It has to be able to perceive these, and then that triggers a series of different signaling pathways. One of these is like the cyclic AMP response pathway, which is responsible for allowing the fungus to build up glycerol concentrations. But the really critical pathway here, which I've highlighted, is this pathway called the PMK1 pathway. PMK1 is a MAP kinase, a mitogen activated protein kinase. So what happens is that surface receptors, such as these two, MSB2 and SHO1, those become activated. They lead to a phosphorylation cascade, And then these three kinases, MST11, MST7, and PMK1, become phosphorylated. And ultimately, they switch on a whole raft of different transcriptional regulators, and that leads to Appressorium development. So in the absence of this PMK1 kinase, if we take this out, and we can do that by targeted gene deletion, so to take out the gene encoding PMK1, then it prevents Appressoria forming at all. So what you get instead is the spore doesn't collapse, sends out a germ tube, and eventually it will just stop growing when it runs out of nutrients. But it's never able to form an Appressorium, and therefore it's never able to infect the plant. So this is a completely non-pathogenic mutant. And that's the first prerequisite to making an Appressorium. The PMK1 pathway has to be activated. We know that what PMK1 is doing is a huge amount of gene regulation. So when the fungus is incubated on hard hydrophobic surfaces where it makes an Appressorium, that leads to more than 3,000 genes. So 3,917 genes, about a third of the genome is differentially regulated in response to that hydrophobic surface. If we look at the subset of those that are regulated by PMK1, then it's about 3,500. Again, about a third of the genome is responsive to hydrophobicity and also dependent on this PMK1 kinase. And in fact, PMK1 itself regulates more than half the genome is differentially regulated in a PMK1 mutant compared to the wild type. And the reason to show that is just that I think it highlights the real complexity of what happens in Appressorium development. You have to turn over about half the genome has to be differentially regulated in order to make an Appressorium. So as well as the transcriptional regulation, we know that the kinase is also directly regulating a whole series of proteins by phosphorylating them. And we've taken an approach using quantitative Phosphorus Proteomics, where we've isolated phosphorylated proteins from the fungus growing on hydrophobic surfaces, either in the wild type or in a mutant lacking PMK1. That's led to the discovery of almost 2,000 phosphoproteins. And we can then start filtering those using targeted proteomics. And that's led us to discover 55 targets of PMK1. Many of these are transcriptional regulators, which become phosphorylated by PMK1 and then ultimately switch on or switch off whole sets of genes. And to give you an idea of the way that works, this is a heat map. This is the mutant PMK1. Here's the wild type G11 on this side. And you can see that there are many proteins which are phosphorylated in G11, but not phosphorylated in PMK1. And they regulate a whole variety of different processes, such as autophagy, such as the cyclic AMP pathway, also the regulation of the cytoskeleton, chitin synthesis, things associated with cell wall biogenesis. And we're now functionally characterizing these 55 different targets of PMK1. So that's the first prerequisite. So the PMK1 pathway is absolutely critical to being able to make an Appressorium. The second prerequisite, though, is the fungus has to regulate Appressorium development in association with the cell cycle. So this is what happens on the rice leaf surface. Here's the spore germinating. There are 3 nuclei, one in each compartment, and you'll see the first of those will divide. See it divided there, and one of the daughters jumped into the developing Appressorium, and the other one went back into the conidial cell. Then they start degrading. So the conidium degrades its first nucleus, then its second, and then its third. So what you can see is that every time Magnaphorta infects, there are initially 3 nuclei in the spore. 3 nuclei become 4 nuclei, and then eventually become 1 nucleus. So it always goes, the pattern is 3, 4, 1. And that happens every single time in every rice blast infection around the world. This is completely invariant. It always happens. So there's always this pattern of 3 nuclei, 4 nuclei, 1 nucleus. And we know that that's the case because there are a whole series of cell cycle checkpoints that regulate this. So that first nucleus has to undergo DNA replication in order for the Appressorium to begin to form. It then has to pass through the G2 phase into mitosis in order for this Appressorium to swell and mature and begin to melanize. And it has to have exited mitosis and again be arrested in G1 in order for the Appressorium to mature and be able to develop pressure. And in fact, a subsequent cell cycle checkpoint, which operates at the DNA replication point, is necessary for the Appressorium then to repolarize. So the whole process is critically linked to Appressorium development. And the third prerequisite is this spore has to die. It has to undergo autophagic cell death. So we know that if in normal situation, if we label the nuclei, here's a nucleus in each of these Appressoria here, the spore has collapsed and died. But if we delete a gene associated with autophagy, so remember autophagy is a process by which organelles and cytoplasm can be recycled. And normally is associated with cell survival when cells consume one cell in order to protect another. What happens is that if we delete a gene associated with autophagy, the spore doesn't collapse now. We've got now 3 distended long nuclei. But the Appressorium, although it melanizes, is unable to repolarize. It can't infect rice. So it's rendered completely non-pathogenic. So those are the 3 prerequisites. They have to happen. Every time an Appressorium forms, PMK1 pathway is activated, the cell cycle control mechanism is activated, and then autophagic cell death occurs. So what happens then? Well, the base of the Appressorium, there's a point called the appressorial core where there's less melanin. So there's melanin, a thick layer of melanin in the dome of the Appressorium. But at this point, where it's in contact with the rice leaf, there's no melanin present. This is the point which will undergo this membrane curvature generation, and then the rigid peg will rupture the cuticle and allow the fungus to gain entry to the underlying tissue. We know that that process involves reorganization of actin. So we can label actin in live cell imaging using LifeAct RFP, and we can see this donut shape, this toroidal network of filamentous actin at the base of the Appressorium that occurs right at the base where the fungus is gaining, is in contact with the plant surface. And the actin itself is scaffolded and held in place by special proteins called septins. So septins are GTPases. They're necessary for repolarization. And you can see that ring forming as the Appressorium develops. And this ring is involved in scaffolding actin. It acts as a lateral diffusion barrier. So polarity determinants, proteins at the center of the cell, which are going to be involved in it being able to rupture the rice cuticle, are held in place by this septin layer. Septins were first discovered in yeast. We know they're involved in cytokinesis, but now we know that they're also critical for invasive growth. The presence of these septins is critical for the Appressorium to work. And indeed, if you delete any of the 4 core septins, there are 4 septins that make that ring. If you delete any of them, then the actin ring doesn't form. You get this tangled mess of actin, and the fungus is unable to infect and is again rendered non-pathogenic. So it can't repolarize, it can't form the normal peg as it would do, and therefore it can't cause disease. So septins are really critical for the Appressorium to be able to function. And what we know is that the septins themselves organize a whole raft of different proteins at the base of the Appressorium. So here are 2 of the septins which form this ring. You can see then they enable this actin reorganization to occur. So we get this donut shape here. Gelsolin, an actin binding protein involved in actin polymerization, forms at the center of the ring, as does the septin kinase, CHM1, RVS167 involved in endocytosis, T1 involved in actin actin polymerization, and XO70 part of the 8-membered exocyte complex required for polarized exocytosis. And those are all required for Appressoria to be able to repolarize and held in place by the septins. What's interesting, though, is we can identify mutants where that doesn't happen. So this is a mutant called SLN1. And you can see that in none of these instances does the correct orientation occur. So in all cases, they're disrupted. Well, SLN1 is an interesting mutant because it's a mutant which actually fails to sense the pressure inside a cell. So SLN1 is responsible for monitoring turgor inside the cell. And when it's unable to monitor correctly the turgor inside the cell, then as a consequence, what happens is that it doesn't repolarize. In fact, SLN1 mutants undergo runaway turgor. They increase in pressure continually. So if we look at a time-lapse movie of the septins in an SLN1 mutant, then what we can see is that rather than forming that beautiful ring structure, we can see that the septins attempt to aggregate and then they degenerate and you get this thin patch of septins forming, but the ring structure doesn't occur. What I hope you can also see is that Appressorium getting darker and darker and darker. And the reason for that is because it's accumulating melanin. So this cell doesn't can't perceive when the appropriate level of pressure is being generated. So it carries on its processes. It carries on generating glycerol, and it carries on melanizing until it becomes hypermelanized. But under no circumstances will ever infect a rice leaf. It can't repolarize. It just undergoes turgor generation, but it can't translate that into physical force because it can't reorganize the septins, for instance. So SLN1 is a turgor sensor that enables that process to occur. And the way that SLN1 works, it binds to stretch-activated gated ion channel proteins. We know that and through its interaction with the protein kinase C cell integrity pathway, that leads to modulation of glycerol formation. So it phosphorylates a protein called phosphodiesterase that downregulates the cyclic AMP pathway, so preventing glycerol formation. At the same time, it phosphorylates the NOx complex, which leads to regulated synthesis of reactive oxygen species. That leads to septin aggregation. And all of these processes occur, leading ultimately to penetration peg formation, all of which is governed by a cell cycle checkpoint. And interestingly, what that really shows us is that the septins themselves act as a scaffold, but really an important signaling hub. So these processes here require this septin aggregation. They regulate septin aggregation itself, but they also enable septin aggregation to lead to many other proteins involved in this repolarization process being correctly organized and correctly configured. So this suggests that septins act as a signaling hub, and this is what we're exploring now in my new ERC grant. We're actually going to try and really explore how do septins undergo this signaling function and act as a platform for so many signaling processes required for Appressorium to repolarize. Okay, so that's what we know about what happens on the leaf surface. What I want to do now is tell you what happens when we look inside a plant. What happens when the fungus is actually now invading plant tissue? And what I think, what I hope you'll see from this is that there are many, many parallels. The fungus is now inside a plant cell, and plants will be actively defending themselves. But many of the processes are really associated and similar. They parallel what actually happens in Appressorium. Okay, so let's have a look inside the cell, the plant tissue. You can see there's the Appressorium on the surface of the rice leaf. If we then are able to pull away, optically remove the plant tissue, then you can see the fungus is forming these bulbous branched hyphae, and they undergo a type of budding called axial budding, and they grow very differently than hyphae do when they're on a surface of an agar plate, for instance. But what's interesting is the fungus is not actually rupturing those cells. So although it's ruptured the cuticle and the cell wall, the plasma membrane of the rice leaf remains intact. So if we make a rice transgenic line in which we've targeted GFP to the plasma membrane of the rice leaf, you can see that the plasma membrane is wrapped around the invading fungus. The fungus is expressing a red fluorescent protein, TD tomato. And you can see that it's bounded by the plasma membrane of the plant. So the rice cell that's invaded is intact and kept intact and is fully viable. And we can see that as the fungus moves from one cell to the next, every time it moves inside a new cell, it's bounded by the plasma membrane. And we call this the extra-invasive hyphal membrane. It's a special compartment, but it's composed of plant membrane. And at the tip of each of those fungal hyphae, you can see a membrane-rich cap. This is, again, a plant structure, but it ultimately becomes a structure called the Biotropic interfacial complex, which is really important in terms of effector delivery. The cell that was initially occupied will then break down. The cell membrane breaks down, and this cell will die. So the fungus will move in a mosaic where it's moving always into living tissue, but the tissue that's left behind will die, and that will ultimately fuel the ability of the fungus to sporulate. And that's what forms those necrotic lesions ultimately. But every time the fungus is growing, actively growing, it's always growing in living plant cells. It's a classic biotrophic in that sense. So heavy biotrophic because it's killing plant cells behind that zone of growth. But in the active zone of growth, it's always moving into living cells. And remarkably, it's always maintaining membrane integrity as it does so. You can see that process, that membrane cap at the base of this invasive hyphae leads to the fungus undergoing a budding process, such that membrane-rich cap that we saw initially becomes what's called the biotropic interfacial complex. The green fluorescent protein structure here is actually in this instance, is an effector which is accumulating in the apoplast. So it's secreted and then accumulates around the germ tube. So it's involved in the suppression of extracellular immunity. So a protein delivered by the fungus into the gap between the plant plasma membrane and the fungal cell wall. So it will inhibit extracellular immune responses. So effectors are basically two different types in Magnaportha. The green effector here, BAS4, is an example of an apoplastic effector. So that will accumulate in the gap between the fungal cell wall and the plant plasma membrane, and it will suppress extracellular immunity. We have many examples of those, for example, SLP1, which will regulate and which will suppress chitin-triggered immunity in the extracellular matrix. But another class of effector are the intracellular effectors. So in this case, this one's called PWL2 and cherry. You can see it in magenta here. This accumulates in the biotropic interfacial complex, or BIC, and that's responsible. Those effectors will move directly into the cytoplasm of rice cells. So those are taken up by rice cells directly into the cells, and we think that involves the stimulation of clathrin-mediated endocytosis in the plant that enables the take-up of those effectors from. But those effectors are delivered from this biotropic interfacial complex, which may start out as being a focal immune response. But that BIC structure is always present, this bright structure where effectors accumulate before they're delivered into rice cells. And there are two different secretory processes, one of which is a Golgi-dependent classical secretion process by which apoplastic effectors are delivered. But the cytoplasmic effectors use a Golgi-independent process, which is still exocyst dependent, and that enables them to be secreted directly into rice cells. The other thing to say about effectors is that there are many effectors produced by the fungus. So Magnaportha produces more than 800 different effector proteins during an infection. So if we do a global transcriptional regulation during a time course of infection from 0 all the way through to 144 hours, you can see that there are effectors which are regulated and expressed very early during infection. There are some which are expressed and peak in expression at 8 hours, some peak in expression at 16 hours, some at 24 hours, some at 48 hours, and some later on during infection, and some both in a bimodal distribution. So we can distribute and classify those effectors into these different modules of gene expression during infection. You can see that in this heat map. Here is a whole group of effectors that comes up very early, another group that comes up later on, module 3, module 4 effectors coming up somewhat later. And most of the classical effectors, the ones that have been identified to date as avirulence gene products, most of those are in modules 4 and 5. Most of those are peak in expression between 24 and 48 hours after infection. But the fungus produces a whole raft of these to suppress plant immunity. The result of this is the fungus overwhelms plant defense. And this is just a snapshot of what happens inside rice tissue. And you can see that the fungus is able to rapidly colonize epidermal and then mesophyll cells, forming this bulbous branched hyphae. Eventually, it will find its way to the vascular tissue, where it forms a much longer, classically cylindrical hyphae. So the fungus, as a consequence of having all of these effector proteins, can rapidly overcome plant defense. It can overwhelm the defenses and very soon occupy much of the plant tissue. So it rapidly colonizes plant tissue. The other thing that this enabled us to do was see for the first time what happens when the fungus encounters a junction between two rice cells. So what we're seeing here is the fungus is in one rice cell, and it's moving into the adjacent rice cell. And as it does so, you can see it swells, and then it undergoes severe hyphal constriction. And you can see that bright green flash, which is formation of gelson and GFP. So what we're looking at there is an actomyosin contractile ring. And that enables the fungus then to move from one cell to the next. And what it's actually encountering there is a pit field. So in each case, those are pit fields. So pit fields, remember, are sites where plasma desmata accumulate, the conduits that enable cytoplasmic continuity between plant cells. And the fungus is encountering those pit fields and is then moving through them. If we look at this at the ultrastructural level, then you can see the extent of hyperconstriction that's actually occurring. So what we're doing here is we're taking slices by transmission EM through that process. So we slice through and make more than 100 slices, and then assemble those and do a tomographic reconstruction of the point of traversal of that cell wall. And what you can see is the fungus has undergone this really severe hyperconstriction down to a diameter of about 360 nanometers, which is the same diameter as a pit field. So it's able to occlude the entire pit field and move through that. And that enables it then to undergo, to maintain plasma membrane integrity as it then enters the adjacent cell. But you can see this really dramatic hyperconstriction that always accompanies cell-to-cell movement. Well, how do we study that process? Well, it's very hard to study that process because most of the signaling pathways which we know are responsible for appressorial development are associated with making appressoria. And if you have a mutant, for example, PMK1, remember the PMK1 mutant, when we delete the kinase, then it will prevent the fungus making an appressorium at all. But what we remembered, I remembered that with PMK1 mutants, even if the mutant spores or hyphae are injected directly into rice leaf, they are still unable to actually cause disease. So that always suggested that PMK1 has another function, not just a function in terms of appressorial development, but perhaps a function inside the plant during invasive growth. So we set out to explore that. And the way we did that was to make a conditional mutant of PMK1. And this is using a process called chemical genetics. So what we do is the following. So we made an analog sensitive PMK1 mutant. So if we make a point mutation in the gatekeeper residue of the ATP binding pocket of PMK1, then what that does, it can change the shape of the ATP binding pocket so that it will now bind to this inhibitor, which is an analog of ATP called NATPH-PP1. This will bind directly to that site. So if we make that point mutation, introduce it into the fungus by allelic replacement, we can then have a mutant where if we add a drug, we can switch the kinase off. And to test it, we did the following experiment. So if we have the normal, the wild type, then we've made this PMK1 analog sensitive mutant, AS mutant. You can see it makes an appressorium completely normally. But if we add the drug, if we add NATPH-PP1, we can then now prevent this fungus from making an appressorium, this mutant from making an appressorium. So we're just eliminating a single kinase, but we're now doing it with a drug. So what that means is that we can enable the fungus to grow to any point of development and then switch off the kinase using the drug. And that enables us then to really understand much more about the biology of this kinase. So then we did the experiment that we'd always wanted to do. So first of all, we enable the fungus to actually infect rice leaves. This is the PMK1AS mutant, but there's no drug. So the kinase is completely normal. What happens is the fungus will fill those rice cells, then it will find the pit fields and it will break out into the adjacent cells. And what I hope you can see is this is a very synchronous process. The fungus is able to build up within those rice cells. It then finds the cortex. It identifies where those pit field sites are. It then undergoes the severe hyperconstriction, and then it moves through into the adjacent cell. And this is what normally happens. So this is what would happen in the wild type, but this is a PMK1AS mutant, but there's no drug present. Now, what happens when we add NATPH-PP1? So now we're going to add the drug. So we're switching off the PMK1 kinase. So when we do that, the fungus will actually fill those cells, and it will fill them up, but the fungus can't get out. It's completely trapped within the cell. So it's unable to move from one cell to the next, totally trapped within that tissue. So what that suggests is that PMK1, as well as regulating appressorial morphogenesis, it's also regulating the ability of the fungus to move from one rice cell to the next. So there are direct parallels between the appressorium and the structure which enables the fungus to move from one rice cell to the next. So you can see that PMK1AS mutants, in the presence of the drug, they're trapped. They undergo this cortical scanning, but they're unable to move into the adjacent cells, that the fungus can't get out of the initial cells, completely trapped within this cell. So, of course, plasmodesmata are the sites of immunity. So there are plasmodesmatal immune responses. For example, in response to reactive oxygen species generation or salicylate production, the fungus, the plant will switch on calosynthates, and it will occlude its plasmodesmata. And it does that to prevent effectors moving from one cell to the next. It does that to prevent viruses from moving from one cell to the next. And remember that viruses, plant-infected viruses, have got movement proteins that enable plasmodesmata to remain open to overcome these responses. So we were interested in what happens to plasmodesmatal immunity responses when we delete PMK1. Well, what we found is that PMK1, in fact, regulates a whole raft of different fungal proteins, which are involved not only in hyphal constriction, but also in terms of effector regulation, potentially manipulating those plasmodesmatal immune pathways. So the experiment that we did to do this was we inoculated the fungus for 26 hours. We allowed it to infect rice leaves. We then, at that point, added the drug, naphthol PP1, to one set of using this PMK1AS mutant. And then we extracted RNA and we did RNA-seq analysis. That showed that there was more than 700 fungal genes are downregulated in the AS mutant in the presence of the drug. Many of those encode secreted proteins, and about 60 of those are temporally co-regulated effectors, which are in these modules 4 and 5. So many of the effectors, which we know are involved in suppression of plant immunity, are regulated ultimately by PMK1. So they're regulated in association with the ability to move from one cell to the next. And again, suggesting how PMK1 is really pivotal to those invasive growth pathways. And similarly, in the same way that we're working in Appressoria, we've extended this analysis using Phosphorus Proteomics. So we've carried out the same experiments we did with RNA-seq, where we've allowed the fungus to infect for 26 hours. We've then added the drug, we've harvested at 32 hours, and then we've undergone phosphorus proteomics to try and identify what are the direct targets of PMK1 during that process. What are the things that PMK1 directly phosphorylates? And what we can find by doing that, we can identify more than 2,700 phosphopeptides. But when we filter those containing a MAP kinase motif and those that are just regulated by PMK1, we can identify 39 potential targets of PMK1, which are associated with cell-to-cell movement. And I'll give you an example of just one of those and the way in which they may work in a moment. But to classify them, many of them are involved in cell division because the whole process, again, is cell cycle regulated. Some of them are involved in transcription factor regulation. Many of them involved in cytoskeletal remodeling associated with that hyphal constriction. But some are involved in vesicle trafficking and also some involved potentially, therefore, in effector regulation. And I'll give you one example of these, one of them that's involved in vesicle trafficking, to give you an example of what we actually see. So here's one. This is SEC31. This is a protein which we identified as being potentially regulated by PMK1, potentially phosphorylated by PMK1. If we look at the distribution of this protein during invasive growth, we can see lots of puncta associated with invasive hyphae. So when the fungus is invading these new cells, there's lots of puncture of SEC31, suggesting there's vesicle trafficking processes associated ultimately with effector delivery, presumably, and delivery of other extracellular enzymes. And we can see those distributed throughout the invasive hyphae. This is in the PMK1AS mutant. So if we then add naphthol PP1 and block the process, what we see is the following. Here's the fungus now trapped inside the cell. But what I hope you can see is that rather than those small puncta, we see large aggregates of SEC31. And you can see that the puncta size, basically the puncta number decreasing in size as the size of them increases. So these aberrant protein aggregates form instead. So what that suggests is that vesicle trafficking is just one of the processes that PMK1 is regulating in order to control this cell-to-cell movement from one cell to the next. So how then is the fungus really moving from cell to cell? Well, again, there are remarkable parallels between the way an Appressorium works and the way the cell-to-cell movement process works, because what we see at the crossing points, at each of those crossing points, what we see is a septin collar. Do you remember the septin, this heterooligomeric structure that we saw at the base of the Appressorium forming that ring structure? Well, we see the same ring structure forming at the junctions between rice cells when the fungus is moving from one cell to the next. And again, to try and identify whether these septins are responsible and required for that cell-to-cell movement, what we did was carry out the same experiment using naphthol PP1 to investigate that process. So if we take the PMK1AS mutant, it's now expressing one of the septins, septin 5, as a GFP. In the absence of the drug, what we can see is that septins are forming at each of these crossing points. You can see them at each of these crossing points, a septin collar forming as the fungus is moving from one cell to the next. However, if we add naphthol PP1, we're now inhibiting PMK1, and what we can see is the septins don't aggregate in the same way. So we're seeing a loose aggregation of them, but there's no assembly into a collar, there's no assembly into a ring structure. So again, suggesting that that process is regulated by the PMK1 MAP kinase. And the septins themselves are responsible for that, for mediating that cell-to-cell movement. So this structure we now call the Transpressorium. So as was mentioned in the introduction, this is a structure which has remarkable parallels to the Aprosorium. So it's a structure formed by magnaportin when it's moving from one rice cell to another rice cell. It forms a swollen structure, very similar to the Appressorium. It then undergoes severe hyperconstriction in the same way as it does when it forms a penetration peg. That enables it to locate a Pitfield site and then move through that into the adjacent cell. But of course, it has to, at the same time, be able to regulate a whole set of different functions, including the suppression of plant immunity, because it's within this hostile environment of the plant cell, where it's having to combat the responses from the plant at the same time. The Transpressorium is not a name that I came up with myself, though. The Transpressorium was first described in 1964, so actually even before I was born. So it was initially described by a group of German scientists who were studying wood rotting fungi, and they noticed these structures associated with movement between plant tissue. And they extended their analysis to a series of plant pathogens, and they said, there's a structure which resembles the Appressorium, which is involved in cell-to-cell movement. And I think they were right. And I think that what we've done is really rediscover this term. And I think it's appropriate to what we actually see in terms of the fungus moving from one cell to the next, the Transpressorium. Okay, so to summarize what is actually happening at this point in the Transpressorium, PMK1 is responsible for two different major types of function. The first is it's able to regulate effector gene expression. We know that there are about 60 effectors that we saw that are regulated by PMK1. Those will suppress host defenses. They'll suppress plasmodesmatal immunity. They'll suppress a whole variety of immune responses. And those are switched on in concert with the ability of the fungus to move from one cell to the next. At the same time, however, PMK1 is undergoing this host sensing response and cytoskeletal remodeling, ultimately leading to septin aggregation, septin ring formation, and that leading to the severe hypoconstriction, actomyosin ring, contractile ring formation, and movement of the fungus from one cell to the next. So this invasive growth pathway. And again, the parallels are remarkable between what's happening here and what's happening here. So the way in which the Appressorium functions, the way in which the Transpressorium functions have some real parallels, both in terms of cell cycle control, in terms potentially even of autophagy, but certainly in terms of PMK1 dependent phosphorylation of a whole raft of different target proteins. And that's what we're now actively exploring. So to summarize everything I've told you then this morning, so magnaportin makes a specialized Appressorium that's involved in infecting rice plants. The reason we study it so much is because, of course, it's critical to being able to control the disease. Appressorium penetration is a septin-dependent process. It requires these morphogenetic septins. It also requires the PMK1 pathway and requires autophagic cell death. A sensor kinase called SLN1 is responsible for sensing once a threshold of TERG has been reached. And only when it's able to sense that will it control the re-establishment of polarity and the and then the septin aggregation and the processes that are involved in penetration peg formation. Tissue invasion requires deployment of a whole battery of fungal effectors, so many effectors, so more than 800 in a typical rice blast genome. Those suppress plant immunity. They may also regulate morphogenesis, but we know many of them are directly involved in suppression of plant immunity. And Magna porta makes an infection structure called a Transpressorium, which enables it to move between rice cells. And that has many parallels between Appressorium, Appressorial morphogenesis. So there are remarkable parallels between Appressorial morphogenesis and between Transpressorial morphogenesis. And those, one of which is the fact that both processes require the PMK1 MAP kinase, which is responsible for regulating the morphogenesis of both of those different infection structures. Okay, so to tell you about some of the people who did the work, the turgor sensing work was all done by this very talented postdoc, Lauren Ryder. Andy Foster developed a lot of methods, CRISPR-Cas9 methods, and studied septins in the lab. Miriam did all of the work on PMK1 dependent transcriptional regulation. Cheyenne identified all of the effectors, that whole effector family. Vincent Ware was involved in all of the work on PWL2 particularly, but he was involved in characterizing effectors. Bozang was involved in looking at the regulation of effectors. Naftali did all of the phosphorus proteomic analysis that I've talked to you about. And much of many of the movies were made by Alice Essaiolo, who's a very talented cell biologist and microscopist who did much of the live cell imaging, supported again by a variety of other students, Camilla and Belaine, who look at invasive growth, and then Clara, who has another project in the lab on lichens, and Iris, who leads all the work on septins. I'm very well supported at the Sainsbury lab. Frank Menke and his team in the proteomics division do a lot of work and collaborate with us extensively on phosphorus proteomics. And my colleagues, Sophie and Camoon, Jonathan Jones, who you heard of in this series before, Matt Moscow, Peter Manas, Cyril Zipfel, and Wembo Ma, all provide a huge amount of inspiration and discussion of this work. And here are some of my external collaborators too, notably Barbara Valence Group in Kansas State. And we're funded by the ERC, but also very generously by Gatsby, who are the funders of the Sainsbury Laboratory. And I'm happy to answer any questions that you've got. Thank you.
Thank you very much, Professor Talbot, for the in-depth talk about elucidating the host pathogen dynamics. I'm sure that our audience would like to interact with you. Some of them have already sent in their questions, which I'll be reading out, and you may please provide your views on them. So, sir, the first question is from Dr. Sudhamay Mandal. Is Magna porta oryzae, the rice blast pathogen, infecting wheat, a case of host jump or something else?
Well, we believe it is a case of host jump. It seems to be a host jump that's come from probably from lowlium infecting isolates. So based on the phylogenetics, there are a number of other forage grasses which are potential candidates, but it's strains of the fungus which are infecting grasses have then made a host jump onto wheat. So it hasn't gone directly from rice blast to wheat blast. It's a grass infecting strain. And that's what is so worrying about the current situation, because the fungus that causes wheat blast has never encountered the one that causes rice blast. They've never coexisted. They've never been in the same geographic region before. And yet we know that in the lab they will cross. So they are fertile and they will undergo sexual reproduction. So so there is the potential for hybridization between them, and that's worrying because they are very separate phylogenetically, evolutionarily.
Thank you. So the other question is from Andre. How do climate changes, particularly higher temperature, impact the spread and development of rice blast?
So that's something of an unknown. I think at the moment we know that I would say with wheat blast there may well be a climate change component to that, because the fungus has tended to be in in the sort of very, the much warmer temperate but more subtropical region. So it's been, Magna porta generally likes to grow above 20 degrees centigrade. So it prefers the higher temperatures. But of course, as those higher temperatures have actually increased in the temperate zones, the fungus appears to be moving northward in the northern hemisphere and south in the southern hemisphere. So in terms of rice blast, though, recent evidence, we've one of my colleagues at Sainsbury Lab, Sophie and Kamoon, his group have done an analysis of European blast populations, and they just went collecting, and they found rice blast much further north than had been previously reported. So they found it in areas even into southern Germany. They found Magna porta isolates on various grasses, suggesting that it is actually moving as associated with climate change. So I think it could be, it could potentially be very serious. And I think that the other thing, of course, as many of you will know, is that after heavy rain, after severe weather events, that's very conducive to blast infections, both wheat and rice. So I think that the heavier rainfall that we're seeing, the really severe weather that we're seeing, is conducive to rice blast epidemics. So I think that we might see more serious outbreaks as a result of that.
Thank you, sir. The next question is from Dr. Arup Mukherjee. Why all the blast pathogens which are able to infect leaf are not able to create neck blast?
Well, that's an excellent question, which I wish I knew the answer to. I mean, I think that what we found is that because the way we generally study blast in the lab, pretty much everyone in the world is working on leaf blast. So people are carrying out assays based on leaf blast because it's harder to do neck blast infections in the lab. But of course, you're right. When we actually translate those findings and look at many of the genes that are implicated in leaf blast are not implicated in neck blast, and vice versa, and we do indeed find that there are some really specialist strains isolates in the field which seem to be neck blast specialists, and we don't really understand those. I think the way in which we're going to have to study those is to do a lot of genomic analysis. So I think that we need to identify what would be really good to do is to do a big GWAS type analysis. I think we're getting to a point now where we can do those much more credibly because we've got so much more genomic information. If we were able to have a whole series of isolates which were neck blast specialists, and we can really see, we can really show that they are much more severe in terms of neck blast compared to rice blast and wheat blast, and vice versa. So those which are really specialist in terms of leaf blast but don't cause neck blast, then we might be able to do some sort of GWAS analysis to try and identify the loci that are involved in that process, and then we can test them experimentally in the lab. I don't think we've been in a position to be able to do that in the past, but I think that we're getting to a point now where we should be able to do that.
Thank you, sir. I'll take the next question from our Madam Director, Dr. Padmini.
Yeah, Dr. Professor Talbot, thank you very much and having an excellent presentation. But I have a small query, just like my other colleagues. I want to know, as we know, generally in the stress field, drought stress field, this blast disease appears very frequently and very densely, as well as nowadays in the climate change scenario, in high temperature, what you told, it is also spreading. So which one is the more sensitive, either moisture stress or high temperature, in your view?
I think it's very difficult for me to say definitively because I've not done the experimental work myself. I mean, I think that I think both are going to be important in that regard. I think that we know the conditions which really favour blast, and it's the frequency which they occur under many of the climate models. They just are going to occur more frequently. So then we're going to see, I think we may well see much more severe outbreaks. I think the other thing, of course, is that under high temperatures, some of the resistance genes themselves break down. So we know that and I know this simply not from my own work, but from reading the literature, that there are examples of where you've got what appears to be fairly robust rice blast resistance, which actually doesn't really work at higher temperatures too. So I think there are going to be some cases there. We need much more resilient rice cultivars where we actually have stacks of multiple resistant specificities, both dominant R genes, but also I think we need the QTL type tolerance, resistance, horizontal type resistance, and also recessive resistance like PI21, for example. I think that the work we've been doing in sub-Saharan Africa, which is based on genomic surveillance, we've been stacking resistance genes, and we're doing all this by conventional plant breeding. So by marker-assisted selection, working with IRI in the Philippines, where we've actually got 3 major R genes, which we know exclude the majority, the overwhelming majority of the prevailing blast population in the area. But at the same time, we never introduce them on their own. We always introduce them alongside PI21, the recessive resistance specificity, and also at least 1 QTL for rice blast tolerance, so that we've got something which is much more durable and robust. And I think that's going to be something that we're going to have to do more and more to try and build some resilience in the face of climate change and the fact that we've got so much more conducive environmental conditions for blast.
Okay, thank you very elaborately you have told. Thank you very much.
Thank you, sir.
Sure.
So the next question is from Dr. Sudhamay Mandar. Your Nature Microbiology 2021 paper states that PMK1 is the central hub in a cascade of regulators that all need to act in concert. Only then can the blast fungus infect plants. Is this finding going to pave way for new interventions to control this devastating disease?
Well, I certainly hope that's the case. I mean, that's certainly the rationale for why we're doing it. I think PMK1 itself is a potential target, but it's actually quite difficult to build a fungicide against such a conserved kinase, which is present in so many different organisms, and where there are homologs, obviously, even in humans and so on, and in plants. So I think that's why, in fact, we were very keen to identify the downstream regulators. So I think that the targets which probably have the most efficacy for us are going to be the transcriptional regulators downstream of PMK1 rather than the kinase itself. It will be the next layer. So we were very, in that paper, we discovered HOX7 as being phosphorylated by PMK1. And I think HOX7 has a really great potential as a target. And the way in which it could be inhibited, it doesn't have to be a chemical approach. It could well be an RNAi approach. So, for example, you can imagine a host-induced gene silencing approach where you target a series of those regulators involved in appressorial morphogenesis using host-induced gene silencing. So that's one way in which I think this could be done using a genetic approach. But also, we demonstrated, again, there was a paper which we published in 2020 in Nature Microbiology, which showed that you can identify inhibitors of septin aggregation, chemical inhibitors, which would prevent rice blast in field scale evaluations, so under field conditions. So we know that if you can prevent septin aggregation, you can really prevent rice blast infection too. Of course, we want to do all we can to get away from chemical interventions, but where chemical interventions can be specific and able to be used at much lower doses than current situations, then I think they're certainly worth considering as well. So we keep an open mind to those. But what we're trying to do, and we're working with various companies on this, is to really develop genetic means wherever we can. So use those targets for other potential RNAi type intervention.
Thank you, sir. So if there are no further questions, we may close this discussion session. So thank you, Professor, for being so patient in answering our queries. We have all benefited from the talk and particularly from the interaction with you. So moving on, I would now like to request Dr. Padmini Swain, Director in RRI, to deliver the presidential address. Over to you, ma'am.
Thank you, Navnita. And I very profusely thank Professor Talbot for a very excellent and excellent lecture, and a good number of people have joined in this platform to hear your topics are very excellent, also very befitting topic for today. That is investigating the cell biology of plant infection by rice blast fungus Magnoporthy. That is a very important plant disease. And I think almost all the rice and wheat workers, they are working on this particular fungus, and many work has been done, but still then so many are still not understood properly. So you have given a very brilliant lecture, and I congratulate you, as well as I thank all the people, those who have participated in the board, virtual board, and all the scientist colleagues from our division, for our institute, from other organizations, universities, scientists, students, and many. I could see our previous director, Dr. D. Maiti. He has also joined. I want to inform you that he is a great pathologist, and he had also, even if superannuated from this institute, but his work is still going on, and he is enthusiastic to join in this program. I thank you, sir. I thank all of you to join in a large number for this particular befitting topic, today's topic. And to say a few, I want to say that as we know, millions of people worldwide depend on rice as a staple food. If a crop failure, this is for any region, poses a real threat of starvation. Rice blast caused by fungus Magnoporthy, particularly this fungus, has been ranked among the most important plant diseases of all of them. Failures of entire rice crop have resulted directly from rice blast epidemics. In addition, the same fungus causes a newly emerging disease of wheat that now threatens wheat production in Bangladesh, you have already shown in your slide, and sub-Saharan Africa. Many of the world's most serious plant diseases are caused by filamentous fungi, such as Magnoporthy. Annually, plant diseases claim up to 40% of the total world harvest, and their control is vital to the development of sustainable agriculture. This is particularly important in face of the climatic emergency, as we need to quickly reduce the environmental impact of our agriculture system while providing more healthy food for a growing human population. All of the plant disease management strategies and techniques that have been generated through research have been brought to bear against rice blast, but often with limited success. Rice blast has never been eliminated from a region in which rice is grown, and single change in the way in which rice is grown, and any change in the way resistance genes are deployed, can result in significant disease losses, even after years of successful management. This disease is a model that demonstrates the seriousness, elusiveness, and longevity of some plant diseases. Rice blast has been widely studied throughout the world. Many investigators have considered to be a model disease for study of genetics, epidemiology, molecular pathology of host parasite interaction, and biology. Recent advances in understanding the genes that govern the avirulence as well as virulence interactions have been made with rice blast, and each advance has helped us to understand how other plant diseases work. It is also important to note that entire genomes of the rice blast fungus and rice itself has been sequenced, and Magnoporthy is the first plant pathogenic fungus to have its genome sequenced and released to the public. The Talbot group is renowned for scientific discoveries, which really have led to new insight into how fungal pathogens evolved the capacity to cause some of the world's most important crop diseases, many of which target staple cereals such as rice and wheat. Because of pathogenic fungi are so difficult to analyze experimentally, we know relatively little about the interaction mechanism of many of the world's most serious cereal pathogens. Magniperthy oryzae shares an important feature with many other plant pathogens, such as rust and powder mildew, which allows them to infect their host. These pathogens used specialized cells called Appressorium, we have seen in your slide, which generate immense pressure to breach the top layer of the plant, outer layer of the plant. Your group recent discoveries have shown that fungal morphogenetic proteins called septins, we have seen how they are working, are essential for rice blast disease. We have seen how the PMK1 dependent septin mediated cell to cell movement by Magniperthy oryzae, we have seen in your slide. They are very essential for rice blast disease. These septins are pivotal to function of Appressorium as well as Transpressorium, which are the infection cells of Magniperthy oryzae that invade host cell once inside the plant. Septins are key determinant of virulence, but are still poorly understood in this context. Professor Talbot's group has shown that PMK1 is the central hub in a cascade of regulators that all need to act in concert. Only then the blast fungus can infect plant. So thank you, Professor Nick Talbot, for resourceful talk. Here I can conclude saying that to rescue the future food supplies, we need new and durable methods of disease resistance, either by deploying the immune system of plant or devising new method to prevent plant infection by pathogens. So your very brilliant talk, I think all my plant pathology friends might have been enriched their knowledge as well as got some clue how to work with this plant disease that is the Magniperthy oryzae. So I thank you immensely again, and I hope all of us together will work for the humanity. Thank you very much. Thank to all of you for your patience hearing and joining in this lecture series of Azadi Ka Amrit Mahotsav. Thank you very much. Thanks to all.
Thank you, ma'am. So it is an honor to propose the formal vote of thanks to conclude today's special talk. We extend our deep gratitude to Professor Nicholas Talbot for accepting our invitation and taking time out from his morning schedule to deliver the special talk. It was a remarkable opportunity to listen to you, Professor, and I hope this is just a beginning of a stronger association with you. I take this opportunity to express our sincere gratitude to our director, Dr. Padmini Swain, for her constant support and guidance to make this program a success. Thank you, ma'am. I'm also grateful to Dr. P.C. Roth, Principal Scientist and Chairman of the Organizing Committee of the Lecture Series at NRRI, for planning and coordinating the activities. I'm thankful to Dr. Sudhamay Mandal, Principal Scientist and Convener of this program, for meticulously planning and organizing the event. We also express our sincere thanks to all the members of the committee. I thank all the heads of divisions, scientific and administrative staff, and the students of NRRI for providing necessary support and for attending the event. I also thank all the scientists and students from other institutes for joining the program and making it a success. Thank you. So that's all for today. Thank you again, Professor Talbot. Stay safe and have a great day.
Thank you so much. Thank you very much indeed.
Okay. Thank you, Dr.
Thank you, Professor Talbot.
Thank you.
Namaste.